Hello and welcome to the State of DM2 Research and Ways to Engage. Excuse me, I'm just gonna pop this over here. This session is part of the DM2 track, and it's intended for individuals living with DM2 and their families. This session is being recorded and will be available for viewing after the conference on the MDF Digital Academy at myotonic.org/digital-academy. I'm Kate Beck, the Special Projects Manager at MDF, and I'm delighted to introduce our presenter today. Dr. Laura Ranum is the founding director of the Center for Neurogenetics and professor of molecular genetics and microbiology at the University of Florida in Gainesville. Before this, Dr. Ranum began her research career at the University of Minnesota as a professor and research director of the Paul and Sheila Wellstone Muscular Dystrophy Center. She has been a member of the MDF Scientific Advisory Committee since 2015. And Dr. Ranum is an internationally known investigator of neuromuscular disorders that result from repeat expansion mutations, such as those that cause myotonic dystrophy type 1 and type 2. Work in the Ranum lab has led to discoveries of the DM2 mutation in 2001, and her more recent work has changed our understanding of how expansion mutations are expressed, teaching us basic lessons about how genes work and paving the way for similar discoveries in other diseases, such as ALS and dementia. So, without further ado, Dr. Laura Ranum.
Thank you, Kate, for the introduction and for the invitation to give this talk. I'm really excited to be here and do this, and I will share my screen. And so I'm going to talk about myotonic dystrophy type 2 today, and also do a lot of comparisons with myotonic dystrophy type 1. And myotonic dystrophy type 2 and type 1 are similar diseases in that they're multisystemic disorders that are caused by repeat expansion mutations. And these expansion mutations are similar in that they're in portions of their respective genes that aren't in traditional protein coding regions. So we'll talk about that. So it was a surprise to us when we learned that mutations like this that are not in protein coding regions can actually encode proteins, and these proteins can accumulate in brains. And so the message that I want to give you today is that unwanted mutant proteins made from these mutations accumulate in DM2 brains and also DM1 brains. And it's really been DM2 that has led the way in our understanding of the brain pathology for myotonic dystrophy and how these proteins are accumulating. So I'm going to tell you how this happens and what we can do to fix it, what we're trying to do to fix it. So, to put this into perspective about how it happens, I need to give you some background in genetics. And so I want you to remember three things: planes, trains, and automobiles. And with the planes, what I'm trying to do is give you a bird's eye view of these genetic diseases and to make connections between myotonic dystrophy type 2, myotonic dystrophy type 1, and other expansion diseases. And with trains, I want you to remember that genes run on both tracks, and that's kind of a new way of thinking about genetic mutations that we need to be aware of. And automobiles. And in an automobile factory, you can think of that as a protein machinery making factory. And what do? You make in a protein making factory, the ribosome, you make proteins. And in an automobile factory, you make cars. But what we learned about the protein machinery is that it doesn't only make cars, it makes unexpected products that are permutations of the original three-letter words. So if we use cars as an example, the other letters are arcs and racks, and these are unexpected products for an automobile factory to be making, and they're unexpected proteins for the ribosome to be making. And I'm going to tell you about that. So genetics 101, an overview of genetics is that our body is made up of many, many different cells, and almost all of the cells in your body have a nucleus and chromosomes. And the chromosomes are how the DNA is tightly packaged. And on these chromosomes, there are different genes, and the genes encode for different sets of instructions. And mostly when we think about genes, we think about what proteins do those genes make. And so if we think about the gene and there's a change in the structure of the gene, that can cause a change in the protein. And so you can see an altered protein here. And that altered protein, if there's a mutation in the protein, could do something it's not supposed to do. And similarly, if you have a change that stops the production of the protein, you could just get rid of the protein, and that would be bad too. Then you'd be missing a protein. So that's kind of how most of the research into how mutations affect people and cells has proceeded with proteins in mind. So the genetic hardware is the DNA, and it's packaged into these chromosomes that I just mentioned. And on chromosome 3, there's the DM2 mutation, and on chromosome 19 is the myotonic dystrophy type 1 mutation. And the DNA is double-stranded, so this is the double strand, the 2 train tracks. There's a sugar phosphate backbone, and then nucleotides that are paired with each other: adenine, thymine, cytosine, and guanine. And these are often abbreviated with the single letter code. And the genomic software is how the code is read. So what happens when the code is used. And so if you open any high school biology textbook, you will see the central dogma. Or if you get on the internet, you'll see the central dogma talks about DNA being made into RNA, being made into protein. And that's true, but things aren't so simple. So in this example here, we have a gene with letters of the genetic code spelled out, and that gene makes a protein, and that protein begins with a methionine initiation codon. And so that's kind of the green light signal for the protein to be made. And the protein adds different amino acids onto it one at a time. They're like beads on the string of a protein. And each amino acid is added when the code is read 3 letters at a time. So that's the word CAR. Just remember, when we used to think about these proteins, we thought that they all had to begin with an AUG initiation codon. And so we were only looking for proteins that began with an AUG or an ATG initiation codon, or this green light signal. But proteins are made by reading the genetic code 3 letters at a time. And so genes are organized in different ways. And if we look in more detail at a gene, you have a promoter region that tells the gene which cells to be active in and which not to be active. So there are some genes that are really active in the brain and other cells that are active in skeletal muscle, for example. And the gene can be divided into different segments. So there's the promoter region, there are exons, and there are introns, and there's something called the 3' untranslated region. And you don't need to remember any of this, except to know that the DM2 CCTG expansion mutation and the DM1 CTG expansion mutation are both in these regions of the gene that when the RNA is made and then put into a protein with the amino acids, those sequences are removed. So they're the parts of the gene that are snipped out before the protein is actually made. And so when microsatellite expansion mutations were first discovered in the early 90s, it was really a puzzle how myotonic dystrophy worked, because the repeat expansion mutation was in this part of the gene that wasn't making a protein. But as research went on, it became really clear that these RNAs themselves can accumulate in cells, and these RNAs have a toxic function when they accumulate inside cells. There are other different expansion mutations that can cause other diseases. So Fragile X syndrome is caused by a repeat expansion that is in the promoter region that leads to a reduction of this protein. So that's a protein loss of function disease. And Huntington's disease is caused by a repeat expansion that's located in an AUG-initiated open reading frame. So this has the signal to make a protein, and this extra long stretch of glutamine amino acids gets incorporated into the protein, and that's thought to lead to then a protein gain of function problem. And all of these things are true, but they're not the complete story. And so when we isolated the gene for myotonic dystrophy type 2 a long time ago, this CCUG repeat looked very similar to a CUG repeat. And we could show that the RNA foci accumulate in cells. So these dots are the RNAs themselves that don't get degraded, but cause problems. And there's a lot of research that I think most of the field has focused on, that focuses on the loss of muscleblind, because muscleblind is a protein that's important in RNA processing that gets trapped in these RNA foci. And then that leads to lots of other downstream changes and maybe the multisystemic clinical features of the disease. So if we, as time went on, in about 2006, this was our view of repeat expansion diseases, that they could be located in promoter regions, in introns that got removed. They could be located in regions of the gene that encode proteins, and like myotonic dystrophy type 1 could be located in the 3' untranslated region, or myotonic dystrophy type 2 in this intronic region. So we thought DM1 and DM2 were really caused by RNA gain of function effects. And my lab was working on a different gene, the spinocerebellar ataxia type 8 gene, and we were really interested in this, especially because it seemed just like myotonic dystrophy, but it caused a brain, completely different brain disease, causes incoordination. And we made a mouse model of this disease gene, and we had human autopsy tissue. And after looking at the mice carefully and the human autopsy tissue, we realized that actually this gene was expressed in both directions. And work in Stephen Tapscott's lab about the same time, a little earlier than our work, also suggested that in myotonic dystrophy type 1, there could be little transcripts that went in the opposite direction. And so this kind of raised questions about the two tracks. And so are genes that are caused by repeat expansions often making transcripts in both directions or not? And the answer is yes, they are often doing that. And then when we were trying to figure out how the SCA8 gene worked, was it more important, the CUG RNA from one strand, or was a polyglutamine more important? Then we discovered that actually those start signals for making proteins are not important, and more proteins can be made from each repeat expansion mutation than were ever expected, even for expansion mutations that were thought not to be able to encode a protein because they had no start codons. So expansion mutations produce unexpected proteins, and in multiple frames. So that means that the 3-letter code to put amino acids doesn't have to start with an ATG initiation codon. It can start one nucleotide over in a different reading frame and produce different amino acid proteins. And so when we discovered this, Tao Zhu in the lab did a simple experiment. He took the ataxin 8 gene that we were looking at in spinocerebellar ataxia type 8, and he expressed that gene, and this version of the gene had the start signal, but a second mutant version that he changed didn't have the start signal. And And this is the measurement of the protein. These bands here indicate that there's a polyglutamine protein there, but the protein was there whether we had that start signal or we didn't have it. It didn't matter. And then that raised a whole bunch of really crazy questions, sort of shifting questions. And that one of those questions is, if there's no need to have an AUG initiation codon, then could the ribosome start translation initiation in the CAG reading frame, but also from an AGC and a frame? And if so, that would make a different kind of protein. It would be different amino acids that are incorporated. And could it make them in the polyserine protein? I'm going to try to get my pointer up here. Okay, so then we had evidence that actually these repeats can make proteins in all three reading frames. And so an analogy that I like to use is that genomic DNA goes to messenger RNA, gets translated into a protein. And so this sentence, the letters are kind of squished together. You can't really read it. The letters are slightly changed when you undergo transcription to produce an RNA. And then when the protein is read, it's read 3 letters at a time. And this sentence is the big red, 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 red dog ran out. And the word red is repeated multiple times. And this has the signal to start the sentence, to separate those words into a sentence, to make a protein. And the discovery of RAND translation was that you don't need this green light signal to make a protein. It's just like in the English language, you don't need to start every sentence with the word the. And in RAND translation, an analogy is you could start right at the red, 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 red dog ran out. That would be one possible way to read this, or you could read it or And so these are scrambled messages. They're different messages. They're unexpected messages in they're different words. They're making different proteins, and they're unexpected proteins. And these unexpected proteins, it turns out, were found we found them in the mice that we made, and we also found them in some myotonic dystrophy cells. And this was reported in our 2011 paper where we discovered RAND translation. But the tools we had to look for these proteins were really limited at the time. And it's taken some years to develop better sets of tools to find them. And I'll talk more about that as we go. So RAND proteins have now been found in 11 different diseases. And you can see these repeat expansion mutations cause different types of diseases. There can be tetra or pentanucleotide expansions. There are lots of trinucleotide repeat expansions. They can be located in normal protein coding regions where you expect to have a protein, but you can still make unexpected proteins in the alternative reading frames. So in the other registers, if you will, of the DNA sequence or in the RNA sequence. And so this has gotten to be, there are quite a lot of diseases with these repeats now. And that's great because there's a lot of research that's gone into these disorders across these disorders, which has really been helpful to understanding the myotonic dystrophies. So myotonic dystrophy type 2, as I mentioned, when we first isolated the gene, we thought this is an RNA gain-of-function disease. And most of the research on myotonic dystrophy still focuses on the RNA gain-of-function effects. But we knew from our work on RAND translation that the myotonic dystrophy type 2 repeats would be making 2 RNAs, and we confirmed that. And the predicted proteins that they would make have these amino acids that are abbreviated LPAC, LPAC, LPAC in the 3 reading frames from the sense RNA and QAGR, QAGR, QAGR from the other strand. We needed to develop tools to detect these proteins, and the kinds of tools that we developed are antibodies that are very specific to these repeats. So these antibodies were easier to develop than the antibodies that were needed to detect the RAN proteins in myotonic dystrophy type 1. So really myotonic dystrophy type 2, this nut was easier to crack with DM2. And so using these antibodies and sections of human autopsy brains that were donated to the research, small slices of tissue that you can put on a microscope slide, we could send in the antibodies that detect the LPAC proteins and then label those antibodies with some lab tricks. And then you get this red signal where the protein is. So where these red dots are is where the LPAC protein is accumulating in these cells in the brain. And you can see different patterns and different brain regions. And they're not all over, but they're tend to be in pockets of throughout the brain. And this is an example of one of those pockets in the hippocampus, which is a region of the brain that's important for memory. There's a region called the dentate gyrus 1 region. And if we look for the RAN proteins in that part of the brain on this autopsy sample, we see that there's a lot of this red staining. So a lot of the RAN proteins are in this area, but there aren't very many in this DG2 region. So this is the same slice of tissue from the same human sample from a DM2 patient. And if we look by using another stain at this tissue, we can see that the tissue that has the RAN proteins in it, it has a bunch of holes in it. And the nuclei of this of the cells, it looks really condensed. And this is a sign of unhealthy cells. If we look at the myotonic dystrophy type 2 region without the RAN protein staining, this looks much more, much healthier. So the cells are round shaped and there aren't a lot of holes in the tissue. The proteins that are made from the opposite strand then are detected with a different kind of antibody. So we had to make a specialized antibody to detect those as well. And we did that and we optimized the antibody, and we can detect RAN proteins from that are made in this QAGR direction as well, from the CAGG repeats at the RNA level in different parts of the brain. And they tend to be in the white matter regions of the brain. And so the white matter regions of the brain are regions of the brain where the parts of the neurons that send signals from one cell to another called axons are located. And the axons are like the wiring system of the brain. And for a wiring system to work, you need to cover the wires, and the wires are covered with myelin, like a myelin sheath. And so you can see this wire is not covered. So this is a very dangerous wire. It's not going to conduct the electrical signals properly. And when we look at control tissue and we don't see the QAGR staining here, the tissue looks like it's intact. When we stain for myelin, we get this deep blue staining, which is an indication that the wires are covered. There aren't holes in the wires. When we look at DM2 regions where there's QAGR staining, the tissue doesn't look as intact, and the myelin staining is really depleted. And so that's an indication that there are white matter wiring abnormalities in DM2 patients. And that's a finding that's been reproduced by multiple labs using imaging and other studies. So it could provide a molecular explanation for the observation that's been made by MRI and other strategies. So what's the connection between RNA toxicity and RAN translation in myotonic dystrophy? there are clearly RNA foci that accumulate, and there are RAN proteins that accumulate. And so when we looked at this in this 2017 Neuron paper, we showed that there's an inverse relationship between the muscle-blind availability levels and the number of RAN proteins. So here, this shows a cells with very tiny RNA foci. As the disease progresses, or some cells have more RNA foci, some cells have just a lot of RNA foci. And if the muscle blind is not sufficient to keep it and trap that RNA in the nucleus, then it can get out into the cytoplasm where the protein making factories are, the ribosomes are, and produce the proteins. And you can see the proteins are labeled in green here, and the RNA foci are labeled in red, and the nucleus is labeled in blue, where if you can, muscle blind overexpression may help to restore muscle blind loss of function RNA, gain of function effects, but it also may reduce RAN protein levels. So, a question that we've had from the beginning of our work has been, are RAN proteins also found in myotonic dystrophy type 1 brains? And to answer this question, we needed better tools. We could detect a few RAN proteins in some of the peripheral tissues, but our antibody tools were just not very good. And it's harder to develop antibodies for the predicted RAN proteins in myotonic dystrophy type 1, because it's a CUG, it's going to make the same amino acid, chains of the same amino acid throughout the protein in different reading frames. So there's a glutamine, a serine, and an alanine in one direction, and a leucine and alanine and a cysteine in the other direction. And so we needed tools to be able to look for some of these proteins. And we were especially interested in tools that might be useful not only for DM1, but also for answering that question in the spinocerebellar ataxias and in Huntington's disease and in other diseases where there are repeats. We knew already about Huntington's disease, but there was a bottleneck in getting the right antibody tools to look for these proteins in the brains of myotonic dystrophy type 1 and other spinocerebellar ataxia cases. And so the new tools that we developed are antibodies that detect polyserine or polyleucine. And this is Monica Banez's work with the help of Eddie Rios in the lab recently. He's a new graduate student that's just joined the lab. But Monica has really been driving this project, and she has shown really nicely that there are RAN proteins shown in red, these aggregates that can accumulate in the gray matter regions. And they can also accumulate in the white matter region that contains this wiring system of the brain, but they're smaller and harder to see. But if you blow up, you can see these little red dots, and these little red speckles are throughout the white matter regions. Polyserine made from the opposite stranded RNA also accumulate, and in the gray matter regions and the white matter regions of the frontal cortex. And there's also a bad electrical wiring system that results from the accumulation of these RAN proteins that accumulate in the white matter regions. And this is the Luxel-Fast blue staining again, and compared to control, where there's really a nice myelination of all of the axons, there are holes in that myelination, and the wiring system is not intact. So myotonic dystrophy type 1 RAN proteins are also found in brain regions that show signs of inflammation. And inflammation is bad. If you hit your hand or you get some kind of abrasion, your immune response will do some good things by creating inflammation to stop the bleeding and repair the wound. But in the brain, you don't really want that. Maybe some aspects might be good, but chronic inflammation is bad, and chronic inflammation is a feature of a lot of diseases with neurological symptoms. And in the regions with these RAN proteins, you can see the accumulation of cells that indicate that the brain is inflamed. So can we improve brain function by getting rid of RAN proteins? And to address this question, we worked in a related on a related disorder because we like the bird's eye view and looking across diseases. And in this particular disease, it's called C9orf72 ALS and frontotemporal dementia. It has many of the same features as the other myotonic dystrophy type 1 and type 2. It makes RNAs in both directions, and it makes RAN proteins, and these are poly GP, GA, GR from one strand of DNA and RNA, and then poly PA, PR, and GP from the other strand. And we used two approaches to get at trying to turn off and reduce the RAN proteins to see what would happen. And the first approach we used was to develop antibodies against the RAN proteins, so to try to just get rid of the RAN proteins directly. And the second approach that we used is to use a gene therapy or small molecule approach, including the use of the FDA-approved drug Metformin, which we showed can reduce RAN protein levels. And we figured this part out after we learned a little bit about the biology of RAN translation, and that there's a key pathway that's important inside the cell. And if we can inhibit that with this FDA-approved drug or a gene therapy that inhibits this Pkr, we decrease the amount of these RAN proteins that are expressed in cells and in mice. And so for the antibody therapy approach, we worked with a company called Neurimmune, and this is just a beautiful image showing antibodies being sent in to target SARS-CoV-2 virus, and you can see in this artistic rendering that these antibodies are really laser focused on attacking the virus. And what we wanted was antibodies that were laser focused on getting rid of the RAN proteins. And particularly, we had one of the antibodies that we made and characterized in collaboration with Neurimmune was an antibody against the GA protein. And earlier, we had generated a mouse model of C9 ALS and FTD that had many of the features of the disease, including the motor neuron loss. And we injected the mice with these antibodies, we being not me at all, but Dr. Lian Wen in the lab, who was a postdoc when she did this work. And she injected these mice in a blinded way. Nobody at the University of Florida knew which treatment group and which was the control group. So we compared affected C9 mice, and we got really excited because it looked like there was a significant difference between the groups. So we didn't know which group was which group, but we were getting very excited and wanted to know. And we were super excited when we were unblinded finally and found out that the GA1 antibody treated mice survived longer than the antibody mice that just got the salt water. And what this antibody did was it reduced the levels of these proteins. It increased the survival of the mice. It improved the behavioral phenotypes of the mice, so how they walked and how what their anxiety level was. It decreased the inflammation that we saw in the brain. And really importantly, it improved motor neuron survival in the spinal cord. And that's important because that's a classic feature of ALS. You lose your motor neurons and then become paralyzed. So a second strategy that we've been exploring is to turn off RAN protein production. We think if we can just turn off the faucet of shut down the RAN translation factory, maybe we could really improve the disease phenotypes. And RAN translation is different than the regular protein translation machinery because it doesn't require this AUG initiation codon. So we thought maybe there's a way by learning about this pathway that we can target RAN translation without messing up the production of all the other proteins that a cell needs. So could we find an Achilles heel of these RAN protein diseases? And as I mentioned, we found this really important link between the expansion RNAs that are made and the activation of a pathway called the Pkr pathway. And when that's activated, that leads to an increase in the production of these RAN proteins. And we figured out a way with gene therapy to reduce the production of those proteins, or with metformin, a really safe FDA-approved drug for type 2 diabetes that seemed to have this additional benefit of reducing the RAN proteins. And some example data from the metformin mouse and cell studies was that a metformin treatment really reduced the levels of the proteins in both the cells and in mice. And it decreased the neuroinflammation that we saw in the brains of the mice. It improved the behavior of the mice. So here's the non-transgenic animals that don't have the gene behave differently than the ones with the gene, and then the drug corrects the behavior. And that's the same as here. There's a difference between how a mouse walks around in this open chamber compared to the non-transgenic animals, the control animals, and then that behavior is rescued with the drug. And again, importantly, we showed that there's a decrease in the number of motor neurons that survive in the C9 mice compared to the control mice. And in the C9 mice treated with metformin, the motor neurons increase their survival. And so the take-home message from both of these studies is that decreasing RAN proteins improves disease without changes to the RNA levels or the RNA foci. And so in C9orf72, we know that the proteins are driving the disease, and getting rid of the RAN proteins is really important. And we're really excited that we were able to start a clinical trial. We're recruiting C9orf72 ALS patients for an open label clinical trial to see if metformin is safe in those patients, and to see if in those patients it reduces the RAN proteins. And we're doing all kinds of fancy things with fancier colleagues than myself to measure the imaging in the brain, and the swallowing function, the careful neurological exams, and the all kinds of molecules that are changed in the C9 patients before and after treatment. And I want to just mention that Guillaume Basset's lab did a beautiful study on metformin and showed that in myotonic dystrophy type 1, in a randomized controlled trial, the group getting metformin had improved mobility. And this paper didn't look at the RAN protein levels, but that's something that Dr. Bazet and I have talked about doing, and we'd love to collaborate and get that done. But first, we think it's important to test this in a mouse model and see if the mice are getting better with metformin. And we have a new mouse model of myotonic dystrophy type 2, so I'm really excited to say that. And this is Avery Engelbrecht, who's giving a poster. So he's talking about that. We have 2 lines of mice where we've cloned a gene from a patient that's donated a blood sample. We made a cell line. We made a bunch of clones of DNA, and we stuck a big piece of the human DNA with a repeat expansion into these mice. And we did this experiment over and over and over, and it took a long time, but we're really thrilled that we have 2 lines of mice now. And that means that each line has an independent insertion of the transgene. And Avery and Elaine and other people in the lab have been breeding these mice like crazy to make many, many copies of these mice so that Avery can have lots of mice to examine. But what we know so far from the small number of animals that he's been able to take down is that these mice, both of the lines, express the transgene, so that's great. They make the RNA. They form RNA foci, so that's great. And they both have about 750 CCTG repeats with some repeat instability, which means we might be able to get a few mice that have longer repeats. And in the future, we have a lot of work to do to characterize these mice and know the biology, to better learn about the biology of the disease. But in the future, if they're a good model of myotonic dystrophy type 2 and they have classic features of the disease, we hope that we can use it for drug tests and to further things, pave the way for future drugs that might be useful for patients with myotonic dystrophy type 2. So as far as therapy development, there are many new genetic tools. There's gene editing capabilities. There's RNA targeting. There's trying to target the repeats and make them contract. And there are efforts to reduce RAN proteins. And each of these has advantages and disadvantages. But I think it's really important to consider these diseases as diseases in which a repeat expansion is making 2 RNAs, and it's making 6 potential toxic RAN proteins. And you might be able to degrade the RNA. And if you do that, you're going to get rid of the proteins that are encoded by that RNA. But you'd only get rid of half of the problem because you wouldn't be targeting the other strand, which could make additional proteins. So we may have to modify things and target both strands, but targeting one strand may actually be enough. We just have to wait and see. And so I like to think of these diseases as things are imbalanced. And in many of these diseases, people get along pretty well for a number of years, and then their problem appears. In congenital myotonic dystrophy type 1, I know that the problems can start much earlier. But in DM2 and C9orf72 ALS and frontotemporal dementia, people go on for decades often without having symptoms of the disease. And so if we think about, we know the mutations cause disease, but if we think about maybe the RNA and the RAN proteins together cause a problem and cumulatively tip the balance to be not healthy. And in other disease, there are these ataxias and Huntington's disease that also make an ATG-initiated polyglutamine disease. So if you think about the myotonic dystrophy mutation, we know that the mutation drives the disease. Without the mutation, you don't have disease. So that's what causes the disease. And for many, many years, we've been thinking that this repeat makes just a CUG, or the DM2 makes just a CCUG. And we know that there's a problem with the RNA foci. And we know that that dysregulates RNA processing, and that contributes to disease. But at the same time, there is an opposite strand transcript. There's a CAG for DM1, a CAGG for DM2, and there are RAN proteins that can be made from each of these reading frames. So you have all of these unexpected proteins. So a really attractive strategy to us is to turn down the production of these proteins, because maybe we could come up with a therapy that would work across many of these disorders. And so finally, to conclude, I want to say MDF funding has been critically important for understanding the causes of myotonic dystrophy and for developing treatments. And my lab has had funding from the MDF, and we're really very appreciative of that. And other labs that are working on myotonic dystrophy have had funding. And I know every every scientist is really appreciative of that. It's really helpful to keep things going. There are therapeutic efforts to target the RNA or protein pathways and repair genes, and to target RAN translation, and lessons from one disease certainly connect to other diseases. And that's what we're excited about. So any gains in one disease are really important for other diseases. And so most of the work that I've talked about today was done by Tao Zhu or Monica Banez or Avery Engelbrecht, but there are many other people in my lab that have contributed to aspects of this work, and collaborators. There are many collaborators at the University of Florida, at Johns Hopkins, at McGill, at Neuroimmune, at Biogen, the University of Bern, and the University of Rochester. And so again, I'd like to thank the Myotonic Dystrophy Foundation and all our funders and you for your attention. And I'm really happy to take any questions that you may have and discuss.
Hi, Dr. Ranum. I'm coming back in here. That was fantastic. And I just want to reiterate that we're open to questions. So please don't be shy, ask your questions. Go ahead and type them into the session chat. And I know it takes a second to formulate questions. So while we're doing that, I can just cover a quick logistical thing. Dr. Ranum mentioned Avery Engelbrecht, sorry if I said that wrong, his research poster. So I posted the information on the chat so you guys can all actually go see that. And we do have the exhibitor showcase again today from 4 to 5. So you can actually go and you can read the research and look at the poster, and also actually ask questions and talk to Avery. So.
And also Dr. Banez is presenting her work on myotonic dystrophy type 1. So she has a talk tomorrow, I think. So
yeah, thank you. I'll find that exact time as well and post it in the chat. So as we wait for questions, Dr. Ranum, I can ask you a question.
sure.
Forgive me if it's a little bit general.
Any questions, great.
DM2 expert. Let's see. What are the barriers to more research for DM2? And again, forgive me if you've covered this a bit, but
no, that's a great question. So, why has myotonic dystrophy type 2 research moved slower than myotonic dystrophy type 1? I think maybe one of the reasons, there are fewer people working on it. So I think we have to get more young people really involved in engaged in myotonic dystrophy type 2 research. So we need to, so I think that this funding of graduate students and postdocs is a great effort by the Myotonic Dystrophy Foundation to engage more young scientists, to keep them in the field and to grow the field. I think another reason is probably because myotonic dystrophy type 1 is typically more severe. There are certainly more severe cases of myotonic dystrophy type 1 than there are of myotonic dystrophy type 2, although myotonic dystrophy type 2 can have devastating consequences when it affects the heart and causes the same kinds of symptoms that adult onset myotonic dystrophy type 1 people have. But what isn't found in myotonic dystrophy type 2 are the congenital cases. And I think that really galvanizes families to fight the disease and raise awareness for the disease and gets additional people interested in the disease. So I think that there's also the myotonic dystrophy type 2 wasn't really recognized as being different than myotonic dystrophy type 1 until the gene cloning era. So it was only really in Charles Thornton's group first described with Richard Moxley and Ken Ricker first described these genetically different people with myotonic dystrophy type 2. And we cloned the gene, about a decade after the myotonic dystrophy type 1 gene was cloned. So, I think there's just more people need to get engaged in this research and move it forward.
Yeah, that's a thank you for explaining that. Getting some more questions now in the chat. I have one from Tom McPeak, and he says more of a statement than a question. I started Metformin in 2014 and noticed that the myotonia in my hands and my difficulty swallowing went away.
Oh, cool. I'd love to, yeah, that's very cool. I think that, it does have an effect. It had that statistically beneficial effect on walking, but I think there's a lot to learn about why, if it will continue to have an effect in a larger study, how it's usually used for type 2 diabetes. And so it will be interesting to see what happens in patients that don't have type 2 diabetes, but in larger cohorts of people, and what effect it's really having on the RAN proteins. Is it really reducing them in parts of the brain, for example? So maybe it'll reduce them in the skeletal muscle too, but we don't know how much. We're still looking at that, or how frequent are they in the skeletal muscle, because we needed to have better tools to look. So I think with the discovery of RAN translation, we didn't have a toolbox. There are great tools to study RNA biology, but they've been built up over decades. And then the RAN proteins were just discovered 10 years ago. And so we have to really build all the tools to look for them. And we're getting better at it, but it's taken some time to look for them. But with metformin, it'll be really interesting. And this is what we're doing for the C9orf72 study, is we're taking CSF samples, and we'll be measuring the RAN proteins in the CSF to see if they're reducing the level of the RAN proteins in the central nervous system, which is really important for ALS. But in myotonic dystrophy, it might be a lot easier to see if you could reduce RAN. If we need better biomarkers, to detect RAN proteins peripherally, and then see what response all the treatments have on RAN proteins. Presumably you want them to go down. I don't think they're good. So there are more questions now, I think.
Yeah, we've got a few more. I have one from Steph. Would you mind explaining what gene therapy is and how it works? I'm having difficulty in conceiving how replacing bad genes in a certain area of the body with good ones can change the genetic structure in other areas of the body.
Yeah, so oftentimes a gene therapy just means that you are you figured out a way to stick a gene which encodes instructions for something into the body. And so gene therapy that can be to replace a missing protein, and oftentimes that's probably the easiest thing to do for a gene therapy. Let's say you don't need very much of an enzyme, but it's really important for your body to function, like to degrade something that is like a lysosomal storage disease. You're missing an enzyme that's really important for keeping this pathology at bay. And so then you don't have to get the gene into everything. You don't have to directly replace the mutation. You just have to deliver a gene that will produce this missing protein. and provide the activity for your cells to work better and clear up the problem. Myotonic dystrophy is a lot harder because there's an expansion mutation that is in every cell in the body, and it's doing something it shouldn't be doing. So it's not like we can just replace and add something that's missing. We have to decrease the activity of that or the effects of this mutation. And so one kind of strategy that you could use, and people are trying to do this, and people have made some headway in model systems with this, is to try to cut out the mutation. So you can deliver a gene therapy that will send instructions to go snip out the expansion mutation. And then, but the challenges with that are you want to send in, if you're going to send in scissors into the brain to snip out a gene, you want them to be super accurate and only target that gene, only target that mutation and trim that back, not some other things. And that's been a challenge. There's a new gene therapy vector that's very exciting that can be turned on and off now. And that was just published in Nature by a colleague of mine, Bev Davidson's lab. And that's combining, that may work really well, because then she can deliver the gene, turn it on when she wants it on, and then take away the drug to turn it back off and get rid of the gene. And so, those are some examples of gene therapies, but it's basically delivering to the body anything that you want to express. And it's just a set of instructions to do something. And some of the instructions nowadays are getting really sophisticated with being able to snip out mutations, but you want to do it in the right place at the right time, get in and out of there. And it's harder to do it when the mutation's everywhere and it's affecting so many different organ systems. It would be easier to block something like RAN translation, or to send in a small molecule that degrades an RNA that can reduce the problem.
And that's, I think we just had a follow-up. Does this delivery system work like a virus from Steph?
Yeah, so it does. So the most common type of gene therapy uses a virus called an AAV virus, or adeno-associated viruses. And a colleague of mine at the University of Florida, who's an emeritus colleague now, he actually discovered this virus many years ago and is started the field of gene therapy. And he used to get the criticism that, why is he studying this virus that doesn't have any toxic effects? It's not important if it doesn't cause a problem. And he saw that if it doesn't cause a problem, that could be a great way to deliver a gene. Then you can replace the viral gene, genetic DNA, with the gene you want to deliver, and so that viruses are definitely used.
Thank you. That's a great explanation. From Lynn Schneider, we have, I think I'm hearing that inflammation increases the RAN protein production and or makes the disease worse. Is that correct? And if so, is that only for the brain, or is it for other systems?
We don't know that inflammation makes the RAN proteins increase, or the RAN proteins make the inflammation. So we're not occur. So we're not sure which comes first, but we do know the RNAs themselves trigger this pathway called the Pkr pathway. That's a viral pathway. And usually when viruses infect the cells, you activate Pkr, and then you change the way you make proteins, because the cell doesn't want to be the virus wants you to make the viral proteins only, and the human body wants you to make the regular proteins. So there's been this sort of arms race between viruses and humans for many years, and we're in one now, I guess, especially acute one, and now with the pandemic. But we think that the RNAs themselves act like viruses and trigger this viral pathway that then changes how proteins are made. And then these proteins can accumulate and cause inflammation. But, and we think that maybe you can also have other insults to the brain that stress the brain and trigger what's called the integrated stress response. And we also know that other parts of the integrated stress response can increase RAND translation. So yeah, we don't, you might have inflammation caused by something else, like a blow to the head or a fall. And oftentimes patients with ALS have some, or ataxia have some sort of injury or some insult that leads to the onset of the disease. And nobody's really ever figured out how they're connected. But we're really interested in that question. We think it's a great question. And, you know, we're Avery Engelbrecht is the postdoc or the graduate student that's working on this mouse is interested in that question. So we may have an answer in the future, but we don't know.
Fingers crossed. Great. From Kim Reynolds. She says, I have DM2 and type 2 diabetes. I take oral metformin. I don't tolerate it well and can only take about 500 milligrams in pill form orally due to gastrosystem gas. Could a metformin injection be more easily tolerated?
That's a great question that I'm not an MD, so it's probably you should direct that to your doctor. I know there's a liquid form of metformin, but I'm not sure. But I think there are other ways to address the stomach upset issues. And we're not, you know, we think that there's a lot more work to do to understand if metformin is going to be good for myotonic dystrophy type 2 patients. But if you haven't noticed any problems other than stomach upset, you know, it might be fine. But I would consult with your, well, you're taking it for type 2 diabetes. So you're not taking it for your disease, you're taking it to control your diabetes. But yeah, you may be able to better tolerate it with food and in a different delivery form. So you should talk to your doctor about that.
Thank you. We have one from Araya. When you mention about measuring RAND protein in the CSF in the C9 ORF, is it in the mouse or the human sample?
Well, from the clinical trial, we are collecting CSF from from human patients, and we're trying to measure it, but we're also trying to measure it in the mice. It's harder to collect CSF from the mice, though.
Just doing a time check. We've got 10 minutes until the end of the presentation, and it looks like we just have a couple more questions, so I think it'll time out well. Is there a concern that the drugs being developed for DM1 and 2 will be harmful to other systems, like liver damage in the long run?
Yeah, I mean, there's a risk for any drug to cause side effects. And so drugs are tested very carefully before they're, especially new drugs, before they're deemed to be safe and effective. I think one of the advantages of metformin is it's got a long track record in as being a generally safe drug. So we're not sure if it's completely safe for patients with these diseases, and that has to be tested, and its efficacy has to be tested carefully. But we know, because people have been taking it for decades and decades and decades, that it's a generally well-tolerated drug. There are some side effects with it, but any drug that you take can have side effects. But by the time it gets to being an FDA approved drug, or even a drug that's being considered in a careful clinical trial, they're going to be monitoring for liver toxicity and side effects like very carefully. So these studies are really well done. And the review process and the FDA approval process is rigorous. So by the time you get a FDA approval stamp, it should be pretty good.
Great. I have a couple follow-up questions, but I'm going to go ahead and try to get everyone's first questions answered, and then we'll loop
Sure.
back if we have more time. Moving down to, are the number of repeats for an individual important to know for DM2? I've heard that the repeats don't mean anything with DM2.
Oh, that's a great question. The repeats in myotonic dystrophy type 2 are really hard to measure because they change in length so much even within a person's blood sample. They're hard to measure because there's so many different sizes. So the normal copy of the gene is a certain size, but the expanded copy can have 75 repeats or up to 11,000 repeats. And we don't have a clear, there's some early papers that suggest that there is a correlation with age of onset, but it's hard to measure the repeats because they're so variable and you can't really get an accurate measure of them. So I think there are some people that tend to have really small ones, and we may learn more about the effect of the repeat with those individuals, but it's just been hard with the human studies. I think maybe we'll get more insight into that with the mouse study. We had the same problem with C9orf72, but it's pretty clear from the mice that we developed that as the repeat length gets longer in the mice with the exact same gene, the disease gets worse in the mice. So we're hoping with the new mice that we have developed that we'll be able to answer that question. But we have a lot of other questions to get to first before we'll get to that question.
She says a few comments and questions. One, is it possible that much of DM2 is not being diagnosed, and so that's why it gets less attention?
Yes, that is very possible that lots of DM2 is not getting diagnosed. And I think that there's more awareness of myotonic dystrophy type 2, but oftentimes if the disease is mild, they may not get an accurate diagnosis of it. And myotonic dystrophy type 1 often comes to the attention of those that have mild symptoms when there's a juvenile onset case or congenital case where the families has to get an answer right away. Whereas in myotonic dystrophy type 2, it's almost certainly underdiagnosed, but the true frequency of the gene through population-based studies should become apparent with time. I know people are working on that.
Thanks. Okay, we're getting to some follow-ups now. So from Lynn, she says, thank you. And a follow-up, it would seem that things that reduce inflammation in the body may help with symptoms then. So her original question was about inflammation.
I think that that's absolutely right, that inflammation is bad and inflammation is going to aggravate things. And I think that anything you can do to reduce inflammation is good. There are lots of books about a diet to reduce inflammation, and I'm happy to send you links if you want to send me an email about books that deal with that topic. There's a lot of sort of an epidemic of inflammation. I think in our society there are lots of people with Crohn's disease, irritable bowel syndrome, different disorders, and people are addressing that with diet is one way to address it. There are also pharmaceutical drugs to address it, but I think part of the reason that we have such an epidemic of inflammation in our society is because of the way we eat. So
How does DM2 impact male reproduction? Excuse me. My wife and I are undergoing IVF and wonder what mechanisms cause the dysfunction.
oh, I'm not sure. That's out of my, I'm not sure about that. Can ask Dr. Day. He should know.
Okay.
Or Dr. Thornton should know.
And Dr. Day will also be doing a stump, the expert DM1, and Dr. Thornton will do it for DM2 tomorrow.
Okay, perfect.
Okay, and then we've got 2 more questions. I think we can cap it off at those last 2, and then we can wrap up after that. That's okay.
Okay.
So we've got 1 follow-up from Araya. Has anyone measured RAN proteins in CSF in either DM1 or DM2 human sample?
Not yet, but we're really eager to do that, and we'd love to do that. We're working on it. Every time you measure RAN proteins in a different tissue, you need to spend a lot of time optimizing the assay. And so it's not like it's super easy to just go from one question to another. There's a lot of optimization, but I think it's a really important question and something we're very interested in doing. So great question.
Great, and it looks like does the number of repeats increase the degree of the disease? So in that case,
I think it is likely to, but we don't have a clear answer for that for DM2.
okay, great. Those are all the questions that we've had submitted.
Okay. Well, I would just like to say that I'd like to thank everybody. First, thanks to the MDF for the invitation. This was really fun, but also I'd like to thank all the DM2 families that we've worked with in the past and that have contributed DNA samples or blood samples, or some people have contributed their brains after they die. And that makes such a difference to the progress that we can make on these diseases. So you guys are the disease experts. We are learning from you, and everything we do, we have to come back to the patients. And so we really appreciate your input, your feedback, and your participation. So thank you.